Hello friends, I am Dr. Sajul Fazi. I welcome you in my YouTube channel. Today in this video, I am going to talk about concept of enzymes. You know friends, enzymes are complex chemicals that control reactions in living cells. They are biochemical catalysts speeding up reactions that would otherwise happen too slowly. Biological catalyst, a molecule which speeds up a chemical reaction but remains unchanged at the end of reaction. Generally names end in AAC, global protein soluble, active site region, cleft or depressions to which another molecules can bind. Substrate fit perfectly and is held in place by temporary bonds which form between the substrates and some of the R groups of the enzymes amino acid. Enzyme-substrate complex. A specific shape of active site will only allow one shape of molecules to fit. So friend, the chemical nature of enzymes. Enzymes are globular proteins. They have a complex tertiary and quaternary structure in which polypeptides are folded around each other to form roughly spherical or globular shape. The overall 3D shape of the enzyme molecule is very important. It is altered, the enzymes cannot bind to its substrates and so cannot function. Enzyme shape is maintained by hydrogen bonds and ionic forces. Enzymes have several important properties. Enzymes are specific. Each enzyme usually catalyzes only one reaction. Enzymes combine with their substrate to form temporary enzyme-substrate complex. Enzymes are not altered or used up by the reactions they catalyze, so can be used again and again. Enzymes are sensitive to temperature and pH. Many enzymes need cofactors in order to function. Enzyme functions may be slowed down or stopped by inhibitors. Friends, biological catalysts, enzymes, typically are very large proteins. Permit reaction to go at conditions that our body can tolerate. Can process millions of molecules every second. Are very specific, react with one or only a few types of molecule substrates. Enzyme molecules naming is easy compared to other compounds. Name is based on what it reacts with, how it reacts, add AAC into. Example, lactase, enzyme that react with lactose. Pyruvate carboxylase removes carboxyl from pyruvate. Nomenclatures, enzyme, substrates or reaction type. Maltase, maltose, urease, urea, proteases, proteins. Urease, urea, proteases, proteins, carbohydrates, hydrases, carbohydrate, lipases, lipids, hydrolases reaction, deaminases, removing amines, dehydrogenases, removing hydrogens. Classification of enzyme based on type of reaction. OXIDOREDUCTASE catalyze a redox reaction. Transferase transfer a functional group. Hydrolase cause hydrolysis reaction. Ligase break COCC or CN single bonds. Isomerases rearranging functional group. Ligase join two molecules. So oxidoreductases catalyze redox reactions. Some common types: reductases, dehydrogenases, oxidases, oxygenases. Look for involvement of coenzymes NAD+, NADP+, PAD and FMN. Transferases, transfer of functional group. Some example: transferases Phosphorolases, transfer of PI or PPI. Kinases, a phosphate transfer, utilizations or generate ATP. Hydrolysis reactions, hydrolases, water molecules used to break bonds. Example, phosphatases, peptidases, APTase and GPTase. Ligases catalyzes reactions that generate double bonds. Add a substrate molecules to double bonds of a second substrate. Examples, decarboxylases, dehydratases and aldolases. Conversions of one isomeric form into another, isomerases. Example, isomerases, epimerases, racemases, mutases. Ligases, two molecules are joined. Anabolic reactions required nucleotides ATP, GTP to drive them. A pyrophosphate bond must be broken. Example, carboxylase and synthetase. Ligases, two molecules are joined. Anabolic reactions required nucleotides ATP, GTP to drive them. A pyrophosphate bond must be broken. Examples, carboxylase and synthetases. Friends, activation energy. What is activation energy? Enzymes increase the rate at which chemical reaction occurs. Activation energy, extra energy temporarily given to substrate to convert it to products. You see the activation energy without enzyme, activation energy with enzyme. Energy of reactant, energy of products, change in free energy, delta T. Now, effect of enzyme on activation energy. For all reactions, you must get over the activation energy hurdle. Like all catalysts, enzyme work by lowering the activation energy for reactions, thus dramatically accelerating the rate of reactions. Most importantly, enzymes are not used up by the reactions. After they have done their work, they release the products and are not. Now friends, effect of enzyme on activation energy. Enzyme change how reactions will proceed, and this reduces the activation energy. It makes it faster. You see, enzyme catalyze reactions. How do enzyme work? Enzyme work by weakening bond which lower activation energy. Total energy, enzyme catalyze reactions pathway. Enzyme lower activation energy for catalyzed reactions, Y with X. Effect of substrate concentration. For non-catalyzed reactions, reactions rate increase with concentrations. Enzyme catalyzed reactions also increase but only to a certain point, Vmax maximum velocity. At Vmax, the enzyme is working as fast as possible. Now friends, you can see substrate concentrations, Vmax, half Vmax, Km. The relationship between the initial velocity of enzyme catalyzed reaction and the substrate concentration. Vmax is the maximum velocity at which the amount of enzyme present can catalyze the reaction, K. So Vmax occurs when enzyme active site are saturated with substrates. K. Michaelis-Menten constant reflect affinity of enzyme for its substrates. The smaller the Km, the greater the affinity an enzyme has for its substrates. Effect of substrate concentration. Vmax, more enzyme, Vmax, some enzyme. Substrate concentration, rate of reaction is. So rate is limited by the concentration of both the substrate and enzyme. Now the active site. The active site has two components, catalytic site and binding site. Model of lysozyme. Characteristic of enzyme active site. Catalytic site where the reaction actually occurs. Binding site area that hold the substrate in proper place. Enzyme uses weak non-covalent interactions to hold the substrate in place based on R group of amino acid. Shape is complementary to the substrates and determine the specificity of enzyme. Sites are pockets of or cleft on the enzyme surface. Now enzyme-substrate reactions. You can see a step in an enzyme reaction. Enzyme and substrate combine to form a complex. A complex goes through a transition state, not quite substrates or product. A complex of enzyme and the product is produced. Finally, enzyme and product separate. All of these steps are equilibrium. Let's review each step. You can see the players: binding site, catalytic site, enzyme substrates. Formation of the enzyme substrate complex. First step in the enzyme catalyzed reaction, enzyme substrate ES complex. Now formation of the transition state and intermediate species is then formed. Formation of the enzyme product complex. The enzyme product complex is then formed. Formation of the product is finally made and enzyme is ready for another substrates. So characteristic of enzyme active sites. Lock and key models. Characteristic of enzyme active site, lock and key models. 1890 pictures by Emil Fischer. This model assumed that only a substrates of the proper shape could fit with the enzymes. Induced fit models proposed by Daniel Koshlands in 1958, and this model assumed continuous change in the active sites structure as substrates binds. Now you can see the log and key models. So induced fit models. These new models recognize that there is much flexible in an enzyme structure. According to the model, an enzyme is able to conform to a substrates. So enzyme classes absolutely specific only react with a single substrate. Group specific work with similar molecules with the same functional group. Linkage specific catalyzes a specific combination of bonds. Stereochemical specific only will work with the proper D or L form. Cofactor and coenzyme. Some enzyme require second species to be present in order to their job. For cofactor type enzyme, apoenzyme and cofactor. Apoenzyme protein portions of enzyme almost ready to work. Cofactor prosthetic groups need to activate the apoenzyme. So cofactor enzyme, you can see substrates, cofactors, non-reacting apoenzymes. Coenzyme, a second unit that temporarily bind to an apoenzyme in order for it to work. So apoenzymes, coenzymes, and coenzymes. So vitamins are often converted to coenzyme. Vitamin B1 coenzyme में thymine pyrophosphate function decarboxylations. B2 flavin mononucleotide carries hydrogen. Folic acid tetrahydrofolic acid amino acid metabolism. Biotin biocytin CO2 fixation. Pantothenic acid coenzyme A acyl group carrier. So factor affecting enzyme activity. Following are the important factors affecting the enzyme activity: temperature, pH, salt and inorganic ion, enzyme concentrations, inhibitors, cofactors and coenzymes. So friends, hope this video will be helpful to you. If you like this video, then subscribe my channel, and thanks for watching.